So this is a lecture for April 20th, 2020 on antibiotic resistance. And the main focus of this lecture is to define some terms associated with antibiotic resistance and then talk about some common mechanisms bacteria use to become resistant to antibiotics, how antibiotics resistance originated, how it's relevant in clinical settings, and then how we can combat antibiotic resistance. And so, as we had talked about in previous lectures, antibiotics are sort of chemicals that can harm or prevent the growth of bacteria. And there are different types of resistance to these antibiotics or chemicals, one of which is known as inherent resistance. And inherent resistance is just the resistance that results sort of intrinsically when a bacterium is resistant just because it lacks whatever the antibiotic is targeting. And so, as an example, there are some bacteria that lack cell walls. They're called mycobacteria. And these bacteria are inherently resistant to antibiotics that target cell wall synthesis because if you don't have a cell wall, it doesn't matter if the antibiotic attacks a cell wall, it won't affect you. But what we think of when we worry about antibiotic resistance in clinical settings is something called acquired resistance, which is a change in the genome or the genetic makeup of the bacteria that converts it from a bacteria that was sensitive to an antibiotic to one that is now resistant to it. And some of the more common antibiotic resistant bacteria are listed here. Two that you're probably familiar with are MRSA and VRSA. They're both versions of the Staph aureus bacterium. MRSA is methicillin resistant, so penicillin resistant, and then VRSA is vancomycin resistant. There are also Enterococcus species or intestinal pathogens that are resistant to vancomycin, and interestingly, the vancomycin resistant Staph aureus or VRSA actually acquired its resistance to this antibiotic from an Enterococcus species. And so bacteria can acquire resistance, or they have different types of mechanisms by which they are resistant to antibiotics. The first is modification of the antibiotic's target. And so most antibiotics target enzymes in the bacteria or structural components such as the cell wall. And so in resistant bacteria, there are often mutated genes that encode these targets. And basically, they remove their ability to act as a target. And so, for example, there are some types of resistance to the antibiotic vancomycin where the bacterium has mutated peptidoglycan subunits or mutated pieces of the cell wall. And the antibiotic can no longer affect these mutant peptidoglycan pieces. And therefore, these bacteria become resistant to the antibiotic. And in some cases, like with erythromycin and chloramphenicol resistance, there are actually ribosomes that become mutated in resistant bacteria. And normally these two antibiotics will bind to different parts of the ribosome and stop protein synthesis. But in a resistant bacteria, they can no longer bind to the ribosome and therefore no longer block protein synthesis. And that makes that bacterium resistant to these particular antibiotics. And so, in addition to modifying sort of the target of the antibiotic, you can modify the antibiotic itself inside the bacterium to make it non-functional or to basically chew it up and degrade it. And this is known as drug inactivation. So any of these modifications that make an antibiotic non-functional or lead to its degradation are mechanisms of drug inactivation. So over here in number 2, you can see that this plasmid for antibiotic resistance is making an enzyme that can actually degrade or chew apart an antibiotic and confer a resistance in that way. And this example here for penicillin resistance is an example of that. So there's an enzyme that's a beta-lactamase or a penicillinase that basically chews up and degrades penicillin and therefore makes any bacteria that express this enzyme, this penicillinase, resistant to penicillin. And alternatively, you can have plasmid for antibiotic resistance that encodes an enzyme that just alters or changes the antibiotic. It might do this by adding a functional group like a phosphate group or an acetyl group to this chemical antibiotic compound and basically render it non-functional. Maybe it makes it unable to bind to its original target. And that's another way or another mechanism of drug inactivation that can make bacteria resistant to antibiotics. And finally, there's a sort of more generic resistance mechanism known as an efflux pump. And it's a pretty simple pump that's designed to pump any unknown compounds or drugs out of bacterial cells. And it does this pretty indiscriminately, so it's not specific for any particular antibiotic. An efflux pump can pump out many different types of antibiotics from the same bacterial cell. And because it's not specific, having the presence of these pumps in a bacterium can lead to resistance not just of one antibiotic, but of many, or lead to what's called multi-drug resistance. And so antibiotic resistance is generally thought of as really bad for humans because we have effectively used antibiotics to treat bacterial diseases. And any resistant antibiotics are therefore, when they cause disease, can't be treated as effectively and can cause a lot of health concerns for us as patients. But in terms of bacteria, antibiotic resistance is actually really beneficial to them. And so if you think about it, if a bacterium has to compete in its natural environment and make an antibiotic to kill off competitors around it, it has to sort of develop some innate resistance to that antibiotic itself, or it might actually accidentally commit suicide and kill itself. And so the development of these genes that helped antibiotic producing bacteria become immune to their own antibiotics are known as immunity genes. And through horizontal genes transfer like conjugation, transformation, and transduction, these immunity genes were transferred from bacteria that produced antibiotics to ones that didn't, and allowed antibiotic resistance to spread throughout bacterial populations and communities. And the way that these most immunity genes and as well as most antibiotic resistance genes are transferred are on plasmids. And we talked a bit about our plasmids earlier in this class, but in our plasmid, the R stands for resistance because this is a piece of DNA or plasmid that has one or more antibiotic resistance genes on it. And our plasmids can be transferred via conjugation, as you can see over here. And so not only can the F plasmids be transferred in this direct contact method, but other types of plasmids as well, including the R plasmids that carry antibiotic resistance. And so you can see these now two antibiotic resistant cells. You've got your light yellow cell here that contains an R plasmid and is making a sex pilus, which can interact with your antibiotic sensitive orange cell that does not contain an R plasmid. And after conjugation, this R plasmid can be transferred to your antibiotic sensitive cell and expressed there. And now since both cells have this R plasmid, they both have antibiotic resistance. And this process can happen really quickly through conjugation or through other horizontal gene transfer methods. And so what I would suggest is that you watch this short video about how this experiment was conducted. But what researchers at Harvard found is that bacterium, when grown on a giant petri dish filled with increasing concentrations of antibiotic towards the center, is that these bacteria on both ends, which did not have any antibiotic resistance at the start of the experiment, were extremely resistant to antibiotics in only 11 days. And so they went from basically 0 to completely resistant in less than 2 weeks. So this process can happen really quickly. And one of the couple things that really causes antibiotic resistance, especially in clinical settings, is the overuse of antibiotics or the overprescription of them, as well as using them irresponsibly. And so normally when you take an antibiotic, there are a high number of bacteria that are killed by that antibiotic and a very few number which are resistant. And if you take your full course of antibiotics, you've efficiently killed every single bacteria that is susceptible to them. You leave very few bacteria left that can receive antibiotic resistance genes from these few orange cells. However, if you don't do that and you leave a couple blue guys behind, what can happen is that the resistant bacteria can multiply themselves because they now have preferential conditions where there's no competitors. And those few blue cells that you've left over can be converted via conjugation, usually, into resistant cells as well. And so the more antibiotic you use in certain situations, the more selection pressure is exerted on this situation. And that can actually lead to even more resistance. And so when antibiotics are prescribed, even when they're not necessary, or when you take antibiotics when you don't actually have particular infections, you can sort of create an artificial selection pressure inside your body that favors the replication of resistant bacteria. And then those resistant bacteria can in turn make others resistant. And so just by kind of using antibiotics when you don't need them, you can create this false sense of needing to evolve by a bacteria, and it will pass on its antibiotic resistance to others, usually, like I said, through conjugation. And this is causing a really huge health problem. And so in the United States alone, there are about 3 million infections and 35,000 deaths every year that are related to antibiotic-resistant bacteria. Globally, this number is even much, much larger, and the cost of treating people with antibiotic-resistant infections is also quite high. And as we've seen, bacteria can evolve antibiotic resistance really quickly, and antibiotic discovery really just can't even keep pace with the evolution of antibiotic resistance. And in addition to the research and development not being able to keep pace anyway, drug companies have actually cut a lot of the funding and research that goes into antibiotics because they're not beneficial from a cost kind of benefit standpoint, right? The cost of developing and discovering a new antibiotic is a lot more than the profits that they will receive by selling it. And so drug companies have cut research into this field. There hasn't been a new antibiotic discovered or on the market for many years. And so how can we combat this problem? And there are several ways to do this, one of which is to fund research into antibiotic discovery, because it is a growing problem. It's a very costly one, not only from a financial standpoint, but from a health and personal standpoint as well. We can also educate people about how to properly use antibiotics, how to not overprescribe them, always to finish your course of antibiotics that you receive, even if you start to feel better. And we can focus efforts on decreasing the use and misuse of antibiotics in clinical settings.